Good evening, ladies and gentlemen. We're just giving a minute or so for people who are still joining. We're going to be with you just now. Okay, right. I see there's still quite a few people joining, but we are going to get going. Good evening. Welcome to another session of the Center for Human Metabolomics National Metabolomics Platform CPD event. My name is Chris Foster, and I will be hosting this event. It is my privilege to introduce our speaker tonight. Dr. Kirkland A. Wilson is an assistant professor in the Department of Genetics and Genomic Sciences at Case Western Reserve University School of Medicine, and a physician in the Department of Genetics at the University Hospitals in Cleveland, Ohio. He is a board certified in pediatrics as well as clinical genetics and genomics. Dr. Wilson earned both his MD and PhD in nutrition from Case Western Reserve University, where his doctoral research focused on propionate metabolism and its relevance to rare metabolic disorders. He completed a combined pediatrics medical genetics residency and subsequently clinical biochemical genetics fellowship at Children's National Medical Center and the National Human Genome Research Institute. An active clinician scientist, Dr. Wilson's work bridges patient care, research, and education in metabolic and genetic disorders. His scholarship includes peer-reviewed publications, textbook chapters, and invited national presentations on inborn errors of metabolism and rare genetic diseases. He also serves as a reviewer for leading journals in the field and has been recognized with multiple awards for research excellence and leadership during his training. Dr. Wilson is deeply engaged in advancing the genetics and rare disease community. He has served in leadership roles with the Society for Inherited Metabolic Disorders, contributed to national training initiatives, and helped develop innovative clinical education programs for medical trainees. His current research interests include improving diagnostic tools for metabolic disorders, developing novel strategies for rare disease treatment, and addressing disparities in rare disease research participation. Tonight, Dr. Wilson is going to talk to us about something that may seem trivial. That is, unless you had to do it, and that is amino acid analysis. So, Dr. Wilson, I'm really looking forward to your presentation, and best of luck.
Thank you. Thank you for that kind introduction. So yeah, I want to talk to you guys tonight about a way that we developed to both detect, separate, and measure amino acids that pairs the realm of cost savings as well as rapidity and the speed by which you can obtain this data and information. And so I know that the audience is made up of a number of different people from a number of different backgrounds and a number of different levels of education. So I want to do some level setting to do a bit of an overview and review to make sure we're all starting at the same page, and then really to dive into the assay itself, as well as to talk a little bit about some of the future directions that my lab is pursuing going forward. And so one thing that I'm sure the vast majority are familiar with is this central dogma where you take DNA, goes to RNA, maybe it goes back to DNA, but once you have the RNA, it goes to protein, and so you have this protein. And then then what? What's the next thing you do? What do you do with that protein? And really that then what is what metabolomics is. Metabolomics is that then what is what the protein does. And at the highest level, metabolomics is where you have proteins, they build stuff, they take stuff, they make it into other stuff, they break it down. And when you talk to a metabolic physician or a metabolic researcher, we're obligated to show this incredibly complex map about all the pathways and how it interconnects, and we scare everybody off and nobody ever wants to do it again. And you kind of put it in the back of your mind and say, I learned that in med school and I'm not going to worry about it anymore. And so while it's true that there are thousands of compounds, pathways, what have you, everybody practices metabolomics. When you're carbo loading, when for a run or a marathon, when you're taking your daily multivitamin, you're affecting these pathways and these processes when you're trying to pick your meal to be healthy versus unhealthy. And then the thing that we're going to focus on in this talk tonight the amino acid metabolism. So if you're taking a protein shake, if you're deciding whether to eat a high protein meal or a low protein meal, to go for that steak versus that salad, that is metabolomics. And at the highest level, metabolomics is the study of all of these compounds, all of these pathways, all of these processes, but specifically in a living system. That's kind of the key difference compared to a number of other ways that people describe metabolomics. It really should be, at the end of the day, in a living system, because that's what you're trying to model and that's what you're trying to measure, that activity, that systemic activity. And so we take this broad swath, but we really try and narrow down onto these small compounds at that small phenotypic level. And specifically, metabolomics is not static. Your metabolic profile, the things that you're studying, the things that you're measuring, they change based off of whether you're sick, whether you're healthy, whether you skipped breakfast or ate breakfast. When you're taking that sample, that metabolic study, that metabolic profile is specific for that time point that you're taking it. And then if you take it multiple hours later, things can and will change. And so that's important to keep in mind as well. But to go through some terminology just to do this level setting, a metabolite is the thing that is involved in metabolism, is involved in that pathway, those chemical reactions. Again, uh, taking being involved in that living organism. An analyte is a term that's often used interchangeably with metabolite, but technically an analyte is a thing that you're actually measuring, whereas a metabolite is anything within that metabolic map. Whereas the metabolome is that entire collection, that entire map, but again, always in a biologic system, always in a living system at the end of the day. And then metabolomics is just that quantification of that entire metabolome, that metabolic map. Anabolism is where you build up something, you create something from a precursor, whereas catabolism is the breakdown of something into product, so that precursor to product relationship. An isomer are compounds that have the same molecular formula, but a different orientation. The classic way that is described is handedness. So you have your left hand and your right hand, and don't overlap. They have four fingers, thumbs, but they don't overlap. And so that's really what they mean by an isomer, where you have the same number of carbons, hydrogens, oxygens, what have you, but in a different orientation. And so we classically say this is D versus L, where the L isomer is for the vast majority of compounds, is what's formed in the human system. And then again, just your metabolic profile is the actual analysis. It's the numbers, it's the measurements that you make for these metabolites, for these analytes. But again, it's always at that specific point in time. And that's why we have to redo these measurements over and over again, because they change based off of how old the person is, whether they're sick, whether they're healthy, how much protein they've eaten, what is that amino acid load. All of that is important. And then you add on the additional complexity of compartmentation, transportation, whether you're getting an organ sample, whether you're doing blood samples, urine, however you collect that sample, all of that plays a role in an effect. And so when you're trying to study and when you're trying to measure metabolomics, when you're trying to do metabolomics, there are two broad camps for how you do this. There's the untargeted approach, and then there's the targeted approach. So when you're doing untargeted analysis, essentially you're saying, I'm going to take a sample and try and measure everything that I can in that sample. I'm not going to target anything. I'm not going to limit anything. I'm going to see what's there and measure what I can. The targeted approach says, from the outset, I'm going to take this sample and I'm only going to measure X, Y, and Z. So in this case, we're only going to measure amino acids, and we're going to ignore everything else that is in that sample. There are pluses and minuses to both. As you can imagine, with the untargeted approach, you get a ton of information. You're not limited. You're not self-limiting, essentially, but it takes more. It takes more technology, it takes better technology, it takes more expensive technology. Sometimes it takes longer. Sometimes it takes more prep work. Sometimes it takes multiple runs, what have you. Whereas the targeted approach, because you know what you're trying to measure and detect from the outset, it's often cheaper and it's often faster. And the ways largely independent of how you're conducting metabolomics. There are shared steps, and the things that really change based off of your approach is really how you're doing the separation. And for our purposes, chromatography is essentially the same word as separation. And then how you're processing the data, how you're interpreting that data. But at the end of the day, for targeted approach, rather than looking at this broad swath of metabolites, rather than trying to look for as many metabolites as possible, you're focusing in on that subset. And at the end of the day, doing a targeted approach, if you know your biochemistry, if you know the underlying chemistry and biology, can be pretty powerful. And it certainly has its utility but specifically when you know what you're looking for and what you're trying to identify. And so we sought to develop both a rapid test, so high throughput, but as well as low cost, specifically for the detection of 40 amines in largely the word amino acid and amines are interchangeable. There are slight differences in the sense that an amino acid has a very specific structure in terms of having an amino group, a carboxyl group, having an R side chain that makes each individual amino acid unique. Whereas an amine is really anything with that amino group, that nitrogen group. And because we can detect things that are broader than just that amino acid label, we just use the more scientific term amines, but they're essentially interchangeable. Um, and so we decided to focus on amino acids because of the importance in a lot of the metabolic processes. Amino acids play a role both in energy, feeding into the citric acid cycle, refilling the citric acid cycle, providing direct energy in other ways, and feed into a lot of different aspects. And it's one of the most common tests that we order from a biochemical standpoint. And so when we look, just to take a step back and to talk a little bit about what these diseases and these disorders look like, again, amino acids are incredibly vital and necessary for health. And again, it's one of the most common tests that we monitor for both health and disease, even outside of your biochemical disorders, your inborn errors of metabolism. Amino acids are known to be important in many diseases, whether it's liver disease and changes in citrulline, or similarly with regards to citrulline, the importance in sickle cell disease or other vascular disorders, because citrulline leads to arginine, and the arginine can increase your nitric oxide and lead to vasodilation. And so you have these secondary knock-on effects as well that broadens the importance even beyond your rare inborn errors of metabolism. But when you think of protein metabolism, at the end of the day, the biggest concern is elevations in ammonia. Because when you have proteins, think amino acids. When you think amino acids, think nitrogen. And when you think nitrogen, think ammonia. And so any step along this pathway, whether it's the whole protein, whether it's the amino acid, whether it's the nitrogen, or whether it's the ammonia itself, are problems involving protein that in the worst case can cause elevations in ammonia, which is directly toxic. And so when you think of amino acid disorders, the classic ones are amino acidopathies. And when you hear that word or those words, really what we're trying to convey are problems removing that nitrogen group. So you have your carbon side and you have your nitrogen side. Amino acidopathies are classically problems removing that nitrogen group. But organic acidopathies often start with amino acids. And so rather than the problem being removing the nitrogen group, the problem is handling all that carbon group after that nitrogen is removed. But there's still amino acid defects. And then urea cycle disorders are problems handling that nitrogen group after it's removed. How do we get rid of that nitrogen group so it doesn't lead to accumulations of ammonia. And so all three of these are broadly what we're talking about when we think of disorders of amino acids. And when you look across the board, when you combine all of these three different broad groups, amino acid disorders affect approximately 1 in 1,000 people or 8 million people worldwide. So a pretty significant population. And again, it includes really any broad enzyme defects, whether it's synthesis, degradation, transport, what have you. All of these things can be affected in these different disorders. And so the way that we typically treat it is through diet restriction. That's the classic thing that you think of for a number of these disorders, limiting protein intake, doing specific amino acid supplementations, formulas so that they get the essential amino acids that they're otherwise lacking in the diet. But this can be hard. It can be difficult to maintain, especially when you add on the additional complexity of a child growing, making sure they have enough essential amino acids and protein to grow and develop, but not so restrictive that you're limiting that growth. And we do know that there are iatrogenic causes from these diets because of secondary deficiencies that can lead to changes in brain development and what have you. One of the most famous cases was a recent study looking at leucine, iatrogenic leucine restriction in patients with propionic acidemia, showing likely effects on intellectual development because of these severe restrictions. And so it's difficult. And that's why we often try and monitor amino acid levels as much as we can in order to minimize that. But also we care about the crisis that happens when you have too much of an accumulation. And so specifically, we call it a metabolic decompensation episode or a metabolic decompensation crisis, where you have this severe and sudden deterioration in homeostasis, specifically in this case related to amino acids, that can be life-threatening, can result in long-term organ dysfunction, again, intellectual delay, developmental delay, at the severe end because of elevations in ammonia or these other analytes, coma, brain edema, seizures, a number of different effects that can be pretty severe and life-altering and life-limiting, to be perfectly honest. And we classically think of the causes for a metabolic crisis as being this catabolic stressor. The prototypic one is surgery. That's a huge stressor on the body. And there are ways that we manage that. So by putting a patient on dextrose, essentially sugar water to suppress catabolism, is the classic go-to, frequent assessments. But you're also worried about infections, fasting, which can be a pretty big problem in our patients because of, to be perfectly honest, a lot of these formulas are not tasty by any stretch of the imagination. So getting a child to take them, it can be difficult. Getting a kid to take them in school can be difficult because of bullying and whatnot. And so that by itself becomes a pretty big challenge. And then at the most severe, having too high of an emotional state is a catabolic stressor. Being too excited, being too afraid, being angry and working yourself up are catabolic states that can be important in your severe disease. But for the ways that we measure and classically monitor amino acids are by one of two ways that are both at the end of the day have to do with column separation. So you put your sample on a column, and that long column allows for sequential separation of your amino acids. And so one of the ways is using ion exchange. And not to get too far into the weeds, essentially you do your separation and then you add on this additional compound called ninhydrin that does a color change, and you're measuring that time and that color change. And that's how you determine which amino acid is which. It requires dedicated equipment, but it's cheap. What you lose with that cheap equipment is time, though. It takes multiple hours, and I'm talking 2.5 to 3 hours per sample. So you have your high quality control, that's 3 hours. You have your low quality control, that's 3 hours, et cetera, all before you have the re-equilibration, you have the calibrations. All of that happens before you get to a sample. So in a day, you're lucky if you run a couple of samples using ion exchange technologies. More recently, in order to get around this, people have been using liquid chromatography paired with tandem mass spectrometry. And again, not to get too far into the weeds, it's mass detection. It's quick. But again, whereas the other one was cheap but slow, this is quick but expensive. And so you have these two ends. And either way, because of the time or because of the expense, you're often limiting your analysis to your large centers, your large laboratories, what have you. And that limits utilization, that limits uptake. And specifically, a number of different organizations, including one of the largest, the Genetic Metabolic Dietitians International Organization, while they recommend frequent and reoccurring amino acids, because again, when you're measuring these metabolites, they change over time. So one measurement, one analysis is not enough. So you have to do this over and over and over again throughout life. They recommend frequent analyses, but then they go on to cite these barriers, whether the cost versus time is limiting access. And so this is what we were targeting. We sought to do something that was both fast but low cost in order to increase availability. But one of the things that people have been running into for huge amounts of time is when you pair these two together, you still have to be able to separate, detect, and measure the amino acids we actually care about in these diseases. And you have to be able to do that in a biologic sample. Because at the end of the day, unrecognized amino acid imbalances are what cause a metabolic decompensation crisis, whether it's because of an illness and a catabolic stressor, or whether it's because of iatrogenic causes. And this is why we need to monitor, and that's why we need to monitor frequently. And so essentially the methodology that we ultimately did develop, we also wanted to make sure that it can be run on a relatively small amount, because when you're doing this often, you're often doing it the most frequently in the smallest populations, in those babies in the first couple of months, who may need to get blood drawn for a number of other reasons. So you don't want to take huge amounts of blood in a patient who can't necessarily tolerate that. And so we got it down to either doing 100 microliters of blood, whether it be blood, plasma, or even lysed RBCs. And we felt that that was important to also test, because RBCs are often one of those things that are left over after you've spun out the plasma and used that, which are often, frankly, just thrown away. And so if you can do this on a sample that doesn't have a lot of other utility, that's an added benefit. But we also tested it in dried blood spots because we wanted to be able to test this in a methodology that a patient can take at home as well, rather than having to come into the hospital, come in to get a lab draw. And so one of the things that's important is to make sure that what you're measuring is accurate and not change because of changes in how you're extracting, how you're prepping, how you're drying things down. And so what we often do in metabolomics is add what's called an internal standard at the beginning. Essentially, you add this in at the beginning, and so whatever changes in that measurement of that internal standard is reflecting the changes that would be from your prep and not from the patient. And we use something called D-alpha aminobutyric acid. Again, remember, the L form is what's formed in humans, and so we're using a D form, something that's not present in humans. And we wanted the prep to ultimately be simple. Yes, we're using an internal standard, but our prep is extremely simple. We take a 3K centrifugal filter, and then we put the sample on the filter and we spin it down. And that's it. You take that filtrate and you run it on the sample, or you run it on the machine. For dry blood spots, we do resuspend first in phosphoric acid. So we put the spot, we do a punch out, we put the spot on the filter, we add phosphoric acid, and then we spin it down and collect the filtrate. But really, that's it. That's our entire prep. So there's no drying down, there's no suspending in methanol or any of these other things. It's a 1 to 2 step process, depending on how you're counting. And so the instrument that we use is what's called a high performance liquid chromatography. And this HPLC is essentially the liquid chromatography that gets paired with tandem mass spec. And so rather than pairing it with the tandem mass spec, which is really the expensive part of that equipment, we pair it with ultraviolet visible light detection. We use a programmable needle to do our derivatization. So essentially, we program the needle to take some of the sample, to take some of the derivatizing agent, which in our case is O-pethaldehyde, OPA, and the borate buffer. It mixes it up in the needle and then it shoots it onto the machine. But there's no reason why you can't do that on a benchtop prep. It's essentially just taking some of the OPA, taking some of the borate buffers so that you maintain the pH, doing a quick shake, and then you would just put that in a vial for the machine to detect, or inject, I should say. We use the C18 column essentially just because it's the most common column used both in clinical labs and research labs. But we did also test it on a C8 column, and the numbers that I'm going to show are different, but it still works. One specific thing that I do want to say is we picked O-pethaldehyde in part because it's relatively cheap. It was cheap because nobody was using it. Because what O-pethaldehyde OPA does is it binds to nitrogen. It binds to the nitrogen groups for our amino acids, but it also binds to the nitrogen that's in the air. So essentially, once you open that bottle, you only had hours to use it before it would all react with the nitrogen in the air and not be usable. So you were essentially wasting entire bottles for not that many samples. But if you know your chemistry, if you know your biology, you can say, I know that this is reacting with nitrogen gas in the air. So if I put it under a noble gas that is non-reactive, we used argon gas, but any noble gas works. The nitrogen gas can't get to the OPA, and it extends the stability for weeks after you open that sample. So knowing your chemistry, knowing your biology at the end of the day plays a huge role when you're trying to do this sort of work. And then we detect at 300, apologies, hold on. And then we detect at 338 nanometers. That's just the wavelength that we detect. Because at the end of the day, just like with the ninhydrin, it's a color change that we're actually measuring that time and color change. And we use essentially two buffers. We have an inorganic buffer and we have an organic buffer. The inorganic buffer is buffer A, the organic buffer is buffer B. And essentially, we start at a high inorganic buffer percentage and a low organic buffer percentage, and then we change. And that's really how we get our separation. The amino acids have different affinities for the organic buffer, and then they come off at different times. And so that gets us to about 41 minutes for the actual detection and separation. And then we added in what's called a carryover reduction phase, where essentially we just add in a bunch of alcohol, essentially alcohol with some of our with some water to just wash off everything else, so that we minimize what gets carried over into the next sample. And then we added in a nice long re-equilibration phase. And so at the end of the day, that's about 53 minutes as compared to the 3 hours for your ion exchange. But it's not as fast as your tandem mass spec, but it's about 10x cheaper. And so that's where we're hitting that middle ground. So not as fast as mass spec, but also not as expensive, but faster than your ion exchange. And so this is what it looks like in our hands. This is what you're actually measuring and detecting. So each one of these labeled peaks is an amino acid, is an amine. You can see the different times. You can see that with this, we use pure standards, all at the same concentration, 100 micromolar. But you can see that the peaks aren't the same size. And this is relatively common when you're doing this sort of detection, because the detection itself is dependent on the amino acid. And so you have to know what you're measuring, how you're measuring, how intense that light reaction, that color change is. But the way that you detect these is to essentially you take each amino acid separately and you take a mix and you shoot it over and over and over again. You change the percentages of your buffers, you change how long the buffers stay around, and you try and get separation between the two. And that's essentially what you do. So if we overlay the different buffer concentrations, you can see that we're going up on one and we're coming down on the other. And then there are two main ways that you get separation. You either get separation through the changes themselves, or what we found is that the vast majority of the separation occurs when you just leave the buffers alone. You just give those amino acids on that column time and exposure to those concentrations, and then they kind of wiggle and fall off, you can think of almost. Um, and if one of the things that you have to do when you're doing this sort of analysis is to look at the assay itself, to define the assay and to define what it looks like. And one of the first things that you have to do is to determine your analytical range. We went from 0 to 250 or 2,500 micromolar, and the R squared for this method was greater than or equal to 0.99 for all analytes. The next thing that you have to do is look at what's called your noise. And so what is that level of measurement, that intensity, that signal, that color change, whatever you want to call it, without any sort of actual analyte being there? And for us with this method, essentially you can think of it as just injecting water with the buffer. But for this method, it was less than 3 micromolar for all of these analytes. And so then you want to look at your limits. Your limit of detection is the smallest amount of that analyte that you can see, and your limit of quantitation is the smallest amount that you can actually measure. There are robust formula and different ways that you can measure and calculate these things. But at the end of the day, we found that our limits were biggest for ammonia, and that was what we expected. Ammonia is pretty labile, it's unstable, it'll evaporate off into a gas in the air. And if you've ever had to measure an ammonia in a patient, you know that there's a number of steps that you have to do in order to preserve that ammonia. Again, showing you our prep, we did none of those things. And so it's expected that our limits were the highest for ammonia. And that's another thing that you want to be able to do when you're doing this sort of assay analysis and validation. You want to be able to explain the things that you find, because if you can't, then that's a gap in the knowledge for the assay, especially if you want to apply that clinically. The less gaps in knowledge that you have, the more robust your assay is, and the less surprises that you'll get when you roll it out from your limited analysis in patients to a broad clinical application where you're seeing thousands, hundreds, millions of samples at a time. And who knows what is going into those samples before and after that you didn't necessarily assess. Um, and then again, as talking about, we put in a carryover reduction phase, but you still have to measure the amount of carryover from one sample to the next. And so you'll do this sequentially across your analyte, your concentration detection range. And so just looking at the 2,500 micromolar sample, we saw that ethanolamine had the largest carryover, a little bit over 29 micromolar, which again makes sense. If you've ever worked with ethanolamine, if you've ever seen it or used it, it's very viscous in the bottle. It's sticky. It comes off almost as a gel. And so just if it's that viscous just in the bottle, it's going to be that viscous on the column and in the sample as well. And so that makes sense that you have that carryover. And then we also saw the second highest carryover in lysine, a little bit over 11. And that also makes sense because if you didn't know, polylysine is essentially a tape adhesive. And so again, if it's sticky, is probably autopolymerizing either in the sample, in the prep, or in the column. And so when you're getting this polylysine, this adhesive stickiness, just like we use it for adhesives in our everyday life, it's sticking on the column, and that increases the carryover. But again, it's important to say that these numbers, almost 30 and 11 respectively, are in 2,500 micromolar sample, way far in excess of what you would typically be seeing physiologically. And then one of the other things that you want to look at is your variability. So your intra-assay variability is the variability between samples. So you take the same sample and you run it back to back multiple times, and you say, what is the variability just from rerunning that sample? What is the variability that gets introduced by the machine? What gets variability introduced by the pump, the buffers, what have you? Because this is a mechanical system. And so when that pump is spinning, maybe you set it so that buffer A is 80% and buffer B is 20%. But with the pump, maybe it was 79.5 or something like that between one run, and then the next run it was 80.3 or something. Those little changes do affect your variability when you're measuring it. Or maybe it was supposed to reach 80% at 15 minutes and it reached it at 14 minutes and 58 seconds or something. These little changes, just from the limitations of the machine, the inaccuracies in the machine, are important to measure because they will affect your analysis clinically. And so for that variability, it was all less than 4 micromolar. And then you also want to look at the inter-assay variability. So you shut the machine down, you go home, you make a new set of buffers the next day, you start up the machine, you do your calibration. What do all of those things affect the variability? Because that is also what it practically looks like in a lab. And again, ammonia was highest. We also saw high glutathione, which is also very labile and unstable. And then we saw trimethylysine, again, lysine, and tryptophan having a high inter-assay variability, but not intra-assay variability. And then one of the other things that we wanted to look at was our sulfur-containing compounds. So specifically, OPA in the solution, it has a sulfur group, a thiol group is another word that we call it. And so we wanted to see what these naturally occurring amino acids that have naturally occurring thiol groups, how that interacts. And this is another reason why it's important to know your biology, to know your chemistry, because what we saw when we looked at this is that those compounds that have single exposed sulfur groups, we could not detect them very well. Cysteine, we couldn't detect at all. Homocysteine, we could at least see, but we couldn't measure it. Instead, what we saw were the disulfide groups, so cysteine and homocysteine. And so essentially all your cysteine is immediately forming a disulfide and going to cysteine. All of your homocysteine is immediately forming that disulfide and going to homocysteine. But when you look at your limits, when you look at your variability, none of that is affected. They're all relatively low. So we know that this happens before we're actually doing the measurement and before we're really doing the detection. But it's important because that cysteine and that homocysteine is not actually cysteine and homocysteine by itself. That cysteine is a combination of cysteine and cysteine. That homocysteine is a combination of homocysteine and homocysteine. And that, again, that's important to know when you're trying to use this clinically, what you're actually measuring. And so when we look at a biologic sample, because again, a lot of all assays start with pure standards, but then you have to take that next step that not everybody does, to look in an actual biologic sample, because pure standards are pure, they're clean. A biologic sample is very much not. And you need to be able to ensure that what you're measuring is the real thing and not some conflicting compound that you didn't even know about because you didn't test. And so we did a number of studies blinded to say, like, how does this compare to a clinical analysis? How does this compare when we're trying to do diagnosis and detection? And so again, blinded, we were able to, this was picked just because of the shock value on most. The disorder is maple syrup urine disease. Essentially, you have elevations in your branched chain amino acids. So that's valine, isoleucine, and leucine. You can see huge elevations compared to the normal control. You can also see that the peaks aren't that nice, sharp shape. So this sample would need to be diluted and rerun in order to get a good analysis, because you do lose some of the concentration when you lose that peak shape. But one of the other things that you see in maple syrup urine disease is the compound alloisoleucine. alloisoleucine, and that's diagnostic. If you see alloisoleucine, that patient has MSUD. And so you need to be able to see and detect that, which we can. We can see it in the disease sample. It's not present in the normal sample. And so overall, this method allows for a single run separation of, again, 40 amino group containing compounds, not actually amino acids, but amines, in under 60 minutes. And again, we're not just looking at amino acids, but we have tripeptides, so glutathione, we have hydrides, ammonia. But one of the limitations is that OPA binds specifically to your primary amino group. And so we do not detect secondary amino groups, proline or hydroxyproline, but those aren't classically associated with diseases. So that was a trade-off that we were okay with making. And we don't detect tertiary amino groups like trimethylamine, which is most common in a disease that causes a fishy smell and isolation. So not that true metabolic crisis. So again, we are okay with making that trade-off. But overall, an extremely simple prep, a simple method that allows for rapid introduction into areas that don't have this alternative analysis. Because again, it's about 10x cheaper than your mass spec, but multiple times faster than your ninhydrin ion exchange. And so just to talk a little bit about future directions, you can't really have presentation without talking about AI and machine learning in 2025. So we are also adapting machine learning and artificial intelligence to metabolomic data. Specifically, we're working to develop a computational AI model that we can apply to inborn errors of metabolism and to these assays that we're developing. Because it is a field that is ripe for low-hanging fruit. When you look at applications of AI and machine learning, which is becoming more and more interchangeable with AI, the application of machine learning to inborn errors of metabolism is severely underrepresented. Even when you just limit it to rare disease, AI and inborn errors of metabolism represent less than 5% of all studies. And just like how common things are common in most areas of research, the common disorders in rare disease, such as cystic fibrosis, are the things that are commonly applied to that they commonly apply AI to. And so inborn errors of metabolism have a number of items and aspects that could benefit from AI applications. It's just nobody's doing it. And it's a shame because as I've been talking about, just with a single amino acid analysis, we're generating 40 different measurements. And so that's 40 different measurements per sample, per patient. And as I've said, you have to do this over and over and over again because the metabolome isn't static. And so if you're doing this over and over and over again, that's a huge amount of data. That's a huge amount of data. And then that's just for amino acids. What about acylcarnitines? What about organic acids that you, again, over and over and over again? So this huge amount of data, AI is perfect about picking out these patterns and doing pattern recognition. And that's kind of what we're already starting to see. When we look compared to control versus just a few diseases, we did 2 patients with maple syrup urine disease, 2 with or 3 with urea cycle disorders, just kind of as a proof of concept. Like, what are the things that we see when we look at this entire analytical profile? And so I just picked out a couple of amino acids, not the full 40, but you're seeing differences, and you're seeing differences in things that aren't necessarily related to the disease. So, for example, 3-methylhistidine is something that's related to muscle health or muscle dysfunction, depending on how you're looking at it. And we see, and neither of those things are directly affected in either MSUD, maple syrup urine disease, or the urea cycle disorders, but there's a difference between these diseases and the control. And then similarly, even if we were to look at amino acids that are related to one but not the other, say ornithine or citrulline, which are important and directly disease related in the urea cycle disorders, but not in the MSUD. I don't directly show this, but there is a difference in the ornithine concentration, even from your urea cycle disorders, but also from your healthy controls, which suggests that there's some sort of dysfunction going on there outside of the disease. So probably some sort of generalized mitochondrial dysfunction. function. And that kind of also bears out when you look at other things like glycine can be a subtle marker for mitochondrial dysfunction. That's different between health and disease. And just doing these like subtle pattern recognitions and pairing that and telling the machine learning model that these are the things that are important, or having it tell you these things are important, can then lead to further analysis. Maybe we need to be focusing more on mitochondrial health, or maybe we need to be doing a better job from a nutrition standpoint if they're having this baseline muscle breakdown that's going on. Maybe these patients aren't actually being optimized from a nutritional standpoint, which honestly is likely because they're not getting the assessments as often as they should. But this is just one example of the way that machine learning and AI can be used to in inborn errors of metabolism to really make it more powerful. And so overall, one of the next things that my lab is working on is to do a similar sort of assay development, but now for acylcarnitines, where we're going to use a similar technology to kind of hit that intersection between low cost but high throughput, but now for acylcarnitines. And so targeting that next that another set of inborn errors of metabolism, classically the ones that have to do with energy and fat metabolism. But again, looking at blood, but also now urine. One of the things that we do for some of these disorders is carnitine supplementation in order to get your toxic analytes to bind to the carnitine and then be excreted out in the urine. But we don't really measure urine from an acylcarnitine standpoint all that often. And so what do the analytes look like in the urine? Can we compare that to the blood? Is there data and knowledge or information there that we can tell in terms of metabolic health that we can then pair with AI to tell us these patterns? And again, developing these predictive machine learning tools to tell us, is this patient in good nutritional health? Are they close to a disease or a crisis? Can we predict a crisis? Because that's not something that we can do right now, by feeding it this large amount of data that as humans we just can't get through. It's just too much to digest, but it's perfect for a machine to go through. And then ultimately applying these methodologies to patient samples for this longitudinal study, or doing these serial analyses, getting it more often. Maybe you can get samples monthly, weekly, rather than twice a year, because it's easier, it's more accessible. Maybe you can discover new biomarkers. Biomarkers are becoming a very important thing in especially clinical trials, as a monitoring method. And so having easier ways to do it, simpler ways to do it, also expands clinical trial access. But with that, I'd just like to thank everybody at my prior institution where I did my training, Children's National, as well as Marshall Summer, who was at Children's National and then went off to start a business on Common Cures, as well as everyone at my new institution, Case Western, Reserve University, and University Hospitals. And with that, I'll take questions. Stop sharing.
Thank you very much, Wilson. To the audience, if you'd like to ask some questions, you're welcome to post that in the Q&A. Alternatively, you can be asked to be unmuted and then you can ask your question in person. While we're waiting, I have a number of questions, and I'll ask some of them and just see how the questions come in. So great method. I can see that there can be lots of applications, particularly in a third world setting. So I was a bit surprised at how good your separation for leucine, isoleucine, and alloisoleucine was, particularly because you're only derivatizing on the nitrogen group, right? So that was quite surprising. Were you surprised by that, or is there an explanation for that, or how do you think about that?
It's very much intentional, and I oversimplified it. It was not easy to get that separation. Okay. Because you're exactly right. We're only derivatizing on that nitrogen group, and they're all branched chain amino acids, so they have very similar chemistry. And it took a long time to get separation between those for that exact reason. We tried to do a sequential gradient buffer change to see if that would work. And ultimately, what we had to do was half step by half step of percentage changes in order to get that separation to see where that concentration lied, that got you separation between isoleucine and leucine. And if you made a half a percentage change, they overlapped. It was very specific and very time intensive and not easy by any stretch of the imagination. And all that's kind of how it works. And so we're picking on your branch chain species, but to some extent, your non-polar species, your neutral species, they all kind of act like this, where you have to do these subtle changes and you just kind of have to keep banging away at it, to get good separation. But one of the things that people tend to do in order to improve separation is to add complexity to the buffer system, so to add a third buffer. And we really didn't want to do that, because we were actively trying to keep this methodology as simple as possible. But it wasn't easy.
Okay. I'm almost glad to hear that you struggled because we struggle a lot. I
Multiple
see there's
years of
a
work. Yeah.
Denise, you have a question?
Yes. So I have a question on the how often do you have to repeat the samples when you see an abnormal sample where the concentration is now elevated for some compounds? How often is it that you have to repeat it? Is it a scenario where you have to auto repeat for all of the abnormal samples where you have to dilute them? And how often do you get it right with the first dilution to actually see all of the compounds? Because with HPLC, it's very easy to, it overlaps and the resolution between those compounds is, yeah, it's very close. So if you do have now an elevated, like the example you showed of the leucine, for example, how will you then still see the ornithine? And so do you go gradually lower with your dilution until you can see everything? Because for some cases, you're going to see that the leucine stays elevated and it does still coelute with the other compounds. So just maybe elaborate on how you guys go about with that.
But for this methodology, we're working on getting FDA approval for clinical utilization. So technically, we repeat all of the samples on a clinically validated test. So far, aside from really leucine, and sometimes phenylalanine, we don't often have to do dilutions because we picked a broad concentration range, but those ones can go outside of that range. And so those are really the two that we've had to do serial dilutions on most often. But those are also the two that clinical assays have to do serial dilutions on, not infrequently. And we do the same process for our serial dilutions as you would do in a clinical assay. But broadly speaking, there are a couple of different ways that you can do it. You can do a stepwise dilution downward. You can, some places, intentionally overshoot or do what they expect to be an overshoot so that you kind of build a window that way and you kind of step up or you step down. There's different ways that you can do it. But you're right, when you do a serial dilution, that at a certain point you're missing everything else. So you can get a good analysis off of that first one, except for those amino acids that are too high. If they do run into other amino acids, and by the time you get the dilution down, that you get back to your detection windows, if you've lost that amino acid, that's when you can play with the separation. If you only care about those 2 or 3 amino acids in that particular window, you can't extend or change the separation. That, to be perfectly blunt, is something that we're going to have to do, and we're going to have to separately validate it. But so far, we've only had that problem with phenylalanine and leucine. And one of the nice things, or I guess one of the lucky things, I should say, is for each of those compounds, the amino acids around it aren't necessarily related to those diseases when we're looking at those high elevations. And so it's lucky from that standpoint in the sense that you can still report out the vast you can report out as all your essential amino acids, all of those naturally occurring non-disease amino acids on that initial run. But if you're trying to get the full 40 analyte profile, if you have to do dilutions, you're right, there's sometimes some extra steps that you have to take that make it a little bit more complicated. But oftentimes when you're worried about a new baby with MSUD, you really just care about what that leucine level is, and everything else can come secondary. So there is that too.
Yeah. Okay, just to add on there as well. So I'm just going to give you an idea of our throughput at the moment at our laboratory, just as an example. So we would run maybe just for serum 120 samples in a week. So given that, you'll have to look at every time you see an elevated leucine, you have to do now serial dilution. So that's why I asked. And then the second question, just linking to that, is the integration with the compounds eluting that close to each other. How long does it more or less take for the integration? Is it like a very intense, because for some of them eluting in the beginning, it's actually really very close to each other.
The biggest barrier as far as your integration is really setting up the programming. A lot of these machines now are pretty good at doing integration, especially if you're trying to get to the baseline. So essentially drawing that line flat. If you set a good retention window, and one of the nice things about this assay is the analytes don't shift very often. And so that's something that you can run into when you're doing chromatography, because maybe the buffer's getting older, the derivative is getting older, or maybe you need to like reprime the system or what have you. You can get analyte shifting a little bit, but in part because of the simplicity, we don't get a whole lot of shifting. So we can set a really tight window saying like, only look for this or measure or do the integration. So drawing that line within this small, like couple tenths of a minute window. And so we get good auto integration just using the baseline programming. And so you just have to set up those tight windows. We do a visual check. So a person is double checking and running behind each of those. But we often don't have to make huge changes, if any changes, because we're able to set such tight windows, which is a good thing, because you're right. Like if you're doing hand integration for each one of these things, a couple samples could take all day. And that's not practical and not the point of this assay. But yeah, because we're able to set such tight retention windows, we can kind of avoid a lot of that.
Okay, thanks.
Just another question from my side. I see the audience is a bit quiet this evening, but maybe you have to do amino acids to appreciate the opportunity. So the cysteine, homocysteine, and I suppose glutathione as well, the sulfur-containing compounds, are they unstable pre-analytically, or does this oxidation happen during derivatization?
So there's some pretty specific environmental, pH conditions and ground conditions that you have to maintain. So it's known that disulfide bonds occur, that the pH can affect the rapidity with which disulfide bonds occur. And so we were kind of limited in the sense that OPA needs a specific acidic pH, and that kind of overlapped with the range in which these things are more likely to cause disulfide bonds. One of the things that we're trying to do to work around that is to essentially add different compounds to the derivatization mix. One of the things that we're playing with is iodoacetate, which essentially binds to the thiol group such that it prevents that disulfide bonds. But then we have to find a new Because the 2-mercaptyl ethanol is the thiol group that we use with OPA that just essentially allows a background for the OPA to bind. It allows for that intermediate binding step. And so it's what we're seeing is that the iodoacetate either still allows the mercaptyl ethanol to function, or more likely replaces the mercaptyl ethanol for that OPA derivatization. So it doesn't seem to affect OPA activity at all. But that's we're essentially going to have to do the revalidation for all of those steps with this addition of iodoacetate. But it does seem like that may be a workaround to protect the cysteine and the homocysteine from going to the disulfides. But I think that happens as part of the derivatization.
Okay, yeah. Yeah, I mean, I think, just from, we certainly know with homocysteine, and particularly in serum samples, it oxidizes. But yeah, in dried blood spots is of course more stable. So I was hoping that, perhaps it's pre-analytical and you would have the stability in dried blood spots. But in this case, it's gonna be the same outcome, yeah.
Yeah, and we specifically looked at that for the same hope, and it's the same in your dried blood spots.
Okay.
That's another reason why we don't think that is happening in the derivatization step and not beforehand.
Okay, and then having the ammonia, that must be quite useful. Even though it's not your best from an analytical perspective, I mean, basically just want to know that it's high, right? So
Is checked for that one as well, because you can get ammonia on this same amino acid assay. And so I wouldn't recommend it if you're worried about hyperammonemia, because this is not faster than just doing essentially an enzymatic assay, which is how you classically measure ammonia. You just stick it in an enzyme mixture and you look at a color change, and that's faster than an hour. Which I would not want to use. I don't want to say that this method should replace that faster test, because when you're worried about an ammonia, you need to know quickly. But if you want to know, like, is a patient chronically elevated just from a baseline amount of ammonia, do you want it? One of the things that we're playing with is I don't want to say stress tests, because nobody does that, but to look at pre and post feeding in patients with urea cycle to see what that catabolic load, or what that anabolic load, what that protein load looks like from both their amino acids, but also for their ammonia. Do they have a transient bump in ammonia? And that is something you can do with one test that I do think is another place that's a wealth of information that you can get with a single assay.
okay, that's very interesting. I don't see questions from the audience, but like I said, I think you need to do amino acids to have questions here.
Yeah.
But I want to thank you for your presentation. It was extremely informative to us, and to the audience. If you don't do amino acids, even you get a glimpse of the complexity of this assay actually, and so that I think is a great learning opportunity. I'd also just like to take this opportunity to thank Sanofi, who's sponsoring these CPD events. And just so that you know, Sanofi has no input into the topics that's selected for the presentations or the content thereof. And with that, Dr. Kirkland, I just want to thank you again for your very insightful presentation. I enjoyed it very much. Thank you, everyone, for attending, and we will see you again next week. And we wish you a very pleasant evening, or if you're in the States, then a very pleasant day, right?
But
thank you, everybody.
chatting with you
Bye-bye.
guys.
Okay. Thank you so much.